The bacteriophages present in the oceans are by far the most abundant organisms there, with concentrations of intact particles as measured by epifluorescence microscopy exceeding their bacterial host by 1 to 2 orders of magnitude, at around 10 to the 9th to 10 to the 10th varions per liter. If we were to count all of these marine phages, we would find around 10 to the 30th total. For sense of scale, this is a number close to the mass of the Sun in pounds. Similarly, even though phages are only around 125 nanometers long, which is about as tall as the wavelength of visible light, if you were to stack them one on top of the other, end to end, you would get a spire that would be around 10 million light-years tall. It would stretch out past the next galactic supercluster. We are finding that these viral agents of bacterial death may be among the most dominant players impacting and modulating marine ecosystems, accounting for 10 to 30% and up to 72% of all bacterial mortality in aquatic systems. All this bacterial lysis involves the release of large amounts of sequestered carbon and other nutrients into the environment as dissolved organic matter and particulate organic matter through a process described as the viral shunt. These released cell contents can then be consumed by heterotrophic organisms specialized at living on non-cell-associated organic matter. Additionally, this process may drive much of the deposition of ancient carbon through the release of large quantities of recalcitrant organic matter, leading to long-term carbon storage in the oceans and the marine snow phenomenon. However, while the viral shunt appears to shift large amounts of carbon annually from marine bacteria into both liable and recalcitrant pools, it also appears to have a profound effect on the efficiency of bacterially mediated carbon fixation. Indeed, the Scanlon and Millard groups at the University of Warwick have found that phage-infected populations of cyanobacteria fix between 4.8 and 2.3 times less carbon respectively than uninfected hosts. Indeed, while they found that phages appear to promote photosynthesis within infected cells, they then appear to use the energy produced to make phage particles rather than fix inorganic carbon into sugars. They estimate that this could prevent the fixation of 0.2 to 5.5 gigatons of carbon every year, or up to 10% of the total produced by the oceans. This is for the kill the winner model, which can be used to explain why the molecular diversity of the oceans is enormous, even though the challenging and stable conditions would seem as if they should select for a dominant and particularly well-adapted species. Indeed, what prevents the most competitive bacteria from globally outcompeting other strains? According to this model, bacterial populations present in the oceans become more vulnerable to phage predation in dense concentrations of clonally related blooms, as bacterial success will naturally lead to the greater success of viral predators capable of infecting them. While there is clear evidence that in some contexts viruses do indeed kill the winner in the case of some bloom-forming species, there are also clear instances in which blooms persist in the presence of infectious viruses. This suggests that a better understanding of host-virus interactions on both a molecular and ecological level may lead to a deeper understanding of environmentally and economically harmful blooms. More specifically, the application of phage therapy to diseased corals afflicted with white plague-like and other diseases has been attempted with apparent success in the Red Sea off of Aqaba and in the Great Barrier Reef off Australia. Indeed, Itad et al. Have found that the application of their phage BA3 can inhibit the progression of white plague-like disease in Favia favis coral, and that treated corals will only infect 5% of nearby healthy corals, rather than the 61% infected by no phage controls. Similarly, Cohen et al. Have isolated a phage infecting Vibrio coralliticus, a pathogen that causes paling due to loss of an animal symbiont, which is capable of rescuing the animal symbiont in vitro.